Hello, and thanks for joining us. Welcome to our CellPress webinar on formation and dynamics of biofilms. I'm Gail Teitzel, editor of Trends in Microbiology. I'll be moderating today alongside my colleague Stephen Matheson, scientific editor at Cell Reports.
Hello, everyone, and thanks for joining us. It's clear now that biofilms can contribute significantly to microbial infections and resistance to antibiotics. In this CellPress webinar, we'll dig into recent advances in the study of how biofilms are formed and look at evolution and trade-offs that occur during biofilm development. We are excited to have three leaders in this field here with us today. Our first speaker, Dr. Thomas Bjarnshult, is a professor at the University of Copenhagen. Our second speaker today is Dr. João Xavier, a principal investigator at the Memorial Sloan Kettering Cancer Center in New York. And our final speaker today is Dr. Erin Mitchell, professor at Carnegie Mellon University in Pittsburgh.
We are truly grateful to all our speakers for making the time to be here, and we'd also like to thank Andor Bitpling for sponsoring this webinar so you all could join us today. Before we jump in, I'd like to remind everyone that we'll have a Q&A session after all three presentations are complete. You can submit your questions at any time during the presentations by clicking on the Ask button at the upper right of your screens. So let's begin. Our first speaker is Thomas Bjarnshult. Thomas is well known for his work on biofilm formation in the context of chronic infections. He has pioneered ways of identifying and visualizing biofilms within infections, as well as developing animal models to better represent what occurs during biofilm infections. Thomas's talk today will focus on in vivo biofilm formation. Thank you for joining us, Thomas. The floor is all yours.
Hello, everyone. So thank you, Gail, and the rest of you. Yeah, so I'll talk to you today about biofilms in vivo. And this is the first slide. We need to get the movie in here. I think it's coming. So obviously there's a difference between the single cells, the planktonic cells that you see here on the left, and also on the biofilms here you can see building up in a flow cell, 7-day time-lapse flow cell confocal microscopy. And you can definitely see the differences with the aggregated bacteria compared to the free-flowing bacteria. And then we just needed to change. Yeah. So biofilms, also to set this afternoon, basically forms everywhere a liquid flows across the surface. It can either be in a creek, on any submerged surface here, submarine, you can see it's cleaned up of biofilm, and also in freshwater pipes. This is a water pipe, possibly 100 years old. You see all this biomass and degradation of the pipe is just slowly clogging up the pipes. And that is in nature, but also when biofilms, they can also form in the human body. Anytime a foreign body, a processes or catheter is inserted into the human body, there's a risk that bacteria and also fungi can attach to the surface and cause problems. And also here, probably everyone is aware of caries and periodontitis, and also all this white stuff when you scrape your teeth after maybe a day without brushing your teeth. That is bacteria and biomaterial forming on the teeth, and can eventually here in this picture cause caries. So the problem is we need another movie in here. Let's wait till it starts. So first of all, the problem is not really that big with planktonic bacteria. And so here's a planktonic bacterium. They can easily be phagocytosed by the white blood cells here in these golf ball-shaped figures. And the white blood cells can fairly easily kill the planktonic bacteria. And also this purple haze representing antibiotics can also fairly efficiently eradicate planktonic bacteria. The problem occurs when fortresses or aggregates of bacteria forms. Then these biofilms aggregates become almost 100% tolerant and resistant both to the antibiotics and also the white blood cells. And yeah, and also here, this is actually a study from 1956. Quite also to set the scene for biofilm infections. So here in I think in human volunteers, they injected 7.5 to the 6 CFU of Staph aureus underneath the skin of these volunteers. Only 50% of these humans actually got infected, and all resolved. Then they took another scenario. They injected only 100 bacteria on an implant just underneath the skin, and all of the human beings that then they got infected, and none of these resolved by themselves. So quick calculations, then planktonic compared to biofilms have 75,000-fold increase in virulence. And of course, in the human body, that's a problem because the bacteria can attach both the tissue, bone, and also in the kidney, and also can colonize any devices, as I talked before. So the classic example, also what I've been working on for the last 8 or 10 years, is cystic fibrosis. Of course, cystic fibrosis is an hereditary disease, and it affects iron pump in all our cells, but especially in the lungs and also in the pancreas. The patients are really affected. In the lungs, it causes a thickening of the mucus. The mucus is normally supposed to entrap bacteria, and then the bacteria can be transported up. But in CF lung, due to this defect in the iron pumps, then the mucus becomes thick, and the bacteria stick in the lungs, and the bacteria cannot be cleared anymore. You get a chronic infection. And here in these pictures, you see a picture, explanted lung from a CF male. He had a chronic Pseudomonas aeruginosa infection for more than 28 years. And this is a fixated lung in formalin. And you can see all these yellow parts. This is where the thick mucus and the bacteria and all the inflammatory cells are gathered. So it's no wonder why these patients actually have a decrease in lung function. Then we take a closer look using a PNA fish. Then we can actually see biofilms, these aggregates of Pseudomonas aeruginosa. Here, this is a specific PNA fish probe. And then in counter stain in blue, we can see the white blood cells, the leukocytes, surrounding the biofilms, but cannot eradicate the biofilm. And also, as in the first cartoon I showed you, this patient actually 4 times a year for 2 weeks, he receives IV antibiotics targeted against the Pseudomonas aeruginosa strain, and also he inhaled colistin and topramycin for the last 20 years. And if we sum up, this patient actually received more than 1 kilo of topramycin, more than 10 kilos of beta-lactam antibiotics, and more than 1 kilo of colistin. So huge amount of antibiotics, and also you can see huge amounts of inflammatory cells, but the biofilm still persists. So what is a biofilm actually? And of course, there's many ways to look at this. I'll look at it from in vivo point of view. But the first pictures here, I think, resembles all biofilms. And this is from a flow chamber. And this is typical biofilm, a lot of aggregated bacteria together. And when we take a closer look, you can see a lot of extracellular matrix. This can be proteins, extracellular DNA, and polysaccharides actually physically binding the bacteria together. And of course, it's in striking contrast to the single cells here just readily attached to a surface. And we made a definition back in 2010 that it's a coherent cluster of bacterial cells. They have to be embedded in the matrix, and they are more tolerant towards most antimicrobial agents and host defense, and that is compared to the exponentially growing planktonic bacteria. So the way biofilms can be studied, for example, in vitro, is using these flow cell systems, and biofilms, bacteria attached to a glass surface and can form, and then it can be get fresh nutrients pumped through, and then it can be observed with a confocal microscopy. And for Pseudomonas aeruginosa, as you saw in the beginning, you can get these very, very kind of beautiful structures. These are referred to as mushroom structures, since they look like mushrooms. But this is in vitro. actually when we look in vivo in infections, we don't see mushroom structures. We only see small molecules. We don't see centimeters after centimeter of biomass. Also, there's an additional layer of host material. Bacteria never just exposed to fresh nutrients. We have the inflammatory cells and host defense in general, and also we have a heterogeneous distribution, so it's small clusters in an entire infection, and surface is definitely not a dependable feature. So you see that in these, this is from a chronic wound, the CF lung, and an experimental implant mouse model. And again, you can see the difference between in vitro and in vivo biofilms. So especially the second matrix and inflammation, I think, plays a big role in the vivo biofilm. So this is from a CF lung. This is an epithelial surface with a small cilia in the bronchi of the CF airways. And this is a mucus plug where the thick mucus and the blue small spots here are the bacteria in enlargement. Here the bacteria are aggregated, but you can see a lot of neutrophils, a lot of pus surrounding the bacteria. Also here in a more closer view, you get the bacteria here in red, specific probe for Pseudomonas aeruginosa, the white blood cells surrounding it, and a lot of dead tissue and dead also neutrophils. And the same we see in a chronic wound, small clusters of bacteria, and this is the surface of the wound. So the bacteria are deeper embedded. They're not up here at the surface exposed to air and fresh nutrients. Then also the second matrix, all this host material across on top of the bacteria. Kind of also can be resembled in vitro. This paper by Forsum et al. From 2010 actually measured the oxygen gradients, and you can see here in this graph, this is an experimental biofilm, and they put an oxygen probe into the biofilm, and when they reached 40 micrometers in depth, then the oxygen almost disappeared. So we did a study in chronic wounds a few years ago, and we could find Pseudomonas aeruginosa very good at denitrifying and very good at anaerobic growth. Actually, as you can see in these measurements, it doesn't really start appearing before it's 40 to 100 micrometers in the wound, whereas Staph aureus, a non-motile bacterium, grows much closer to the surface. So the second matrix is very important if you want to study biofilms, to take into consideration where the bacteria actually are situated. And also, you can also find bacteria not attached to a surface, and also to show that they get actually the same phenotypes. We made an experiment here. We grew biofilms in flow cells on a surface, and also suspended in media in these 6-well plates. And just by looking at the scanning electron microscopy here, we see aggregated bacteria, and it's the same kind of aggregation, the same host material actually attaching the bacteria together. And I'm not going to show it here, but the antibiotic tolerance, also the tolerance towards the host defense, is 100% the same. And also, just because bacteria attach to a surface, then we need another movie in here. Doesn't necessarily mean that the white blood cells cannot eradicate or phagocytose the bacteria. Let's get it started. Oh. So you can see the white blood cells here moving over the surface, and this is a single layer of Pseudomonas aeruginosa. So you actually see the white blood cells, the leukocytes, can phagocytose, can eat the bacteria right off the surface. So surface attachment does not protect bacteria from the host defense. Let's see, get the next. Oh, and then we need another movie. But size itself does not matter either. So it doesn't matter if biofilms are big, because the white blood cells can still eradicate them. So here are the white blood cells again. And here we have a quite big aggregate of bacteria. And then you can see this phagocyte, this leukocyte, starts to phagocytose it. It's too big for it to eradicate. But then one of its friends, these are human leukocytes, comes along and also takes a bite of the biofilm. So they can eradicate quite large aggregates. But what actually protects the biofilms in vivo? So we need another movie in here. So again we isolated human white blood cells, and this time we took some supernatant from experimental biofilm and added a live dead stain. So everything which is green is alive, and if something turns red, it starts to die. And you can see in this movie, this is time course for 10 to 15 minutes, that the leukocytes after around 10 minutes starts to become red, and due to osmotic pressure actually swells up, and within 15 to 20 minutes all the leukocytes are gone. So what actually protects at least the bacteria in the biofilms? For Pseudomonas aeruginosa, one of the protection mechanisms is the production of rhamnolipid, which we purified some years ago and explained as to be this protective mechanism against PMNs and polymorph nuclear leukocytes. And we also tested that in an implant mouse model. So we used hollow tubes, silicone tubes, which we pre-coated with Pseudomonas aeruginosa, inserted into the peritoneal cavity of the animals, and then we sacrificed the animals at day 1 and 2. And this is day 1 post-insertion of the implants. So we cut the tube in half, and this is the inner lumen of the tube, scanning electron microscopy. And this is a kind of the army of white blood cells crawling in from here to outside, eradicating all the bacteria on its way in. You still see an intact biofilm. This is again day 1. The neutrophils looks fairly round shaped and healthy. And also you can see here they're phagocytosing the bacteria. Even closer look, see the same. What happens? They crawl across the surface, eradicating all this immature biofilm. But then on day 2, the picture was completely different. Here we have a lot of bacteria, and suddenly the neutrophils are not looking that healthy anymore. They're all full of holes. So here they've been lysed due to this rhamnolipid, and you can see here all the degraded neutrophils, and the biofilm just gets thicker and thicker. So in summary, from this from research we've been doing, also many others, biofilms definitely play a role in chronic infections. And we just started to unpeel or to peel the onion to start to peel what is a biofilm and its role in chronic infections. We know a lot about it, but we still need to do a lot more in diagnostics. We need to do a lot more in treatment, because biofilm infections are really untreatable as it is right now. Now, most important, we need to be able to prevent these infections to actually take place. So I'll round this up with my funding sources, of course, very important. And this is my group in Copenhagen and my collaborators, done all this job for many, many years. And just last thing, so we actually run an online course in biofilms in chronic infections. I'm not going to take more of your time, but you can go to this website, biofilmcourse.ku.dk, and we run courses every fall, and a new one will start next fall in 2014. So by that, thank you, and looking forward to all the questions.
Well, thanks very much, Thomas. Really interesting. Wish you could hear us applauding, but I guess that's not how a webinar works. We'll move on now to hear from our second speaker, who is João Xavier. João is a computational biologist, and his research focuses on the complex interactions that occur during group behavior in collections of cells. Today, he's going to be talking to us about evolution of hyperswarming in experimental bacterial populations. João, thanks very much for being with us.
Well, thank you, Stefan. Thank you for the invitation, and it's great to follow up on Thomas, because he actually introduced Pseudomonas aeruginosa and biofilms in such a beautiful way. And now I'm going to talk about experimental evolution in Pseudomonas aeruginosa. So this is work that we just published very recently in the journal Cell Reports. Came out last month. So as I said, Thomas introduced our player here today, which is Pseudomonas aeruginosa. It's an opportunistic human pathogen, and it's well known for its chronic infections of cystic fibrosis patients. It's also known to evolve in the course of a chronic lung infection, which can take up decades. And we're very interested in understanding how different selective pressures drive the evolution in this pathogen, and especially to look at this at the molecular level. We study mainly a collective trait of Pseudomonas aeruginosa that's called swarming motility, which is a behavior by which the bacteria can travel in groups across soft surfaces. And here we're going to play the movie Wild Type. Okay. Let's see if we can get this started. Okay. So this movie is a time lapse that shows how Pseudomonas aeruginosa swarms. You see these tendrils shooting out at the center of the colony, and they travel towards the edge of the plate, and they bifurcate along the way. It's a very interesting example of collective behavior because these bacteria, as single-celled organisms, they couldn't travel by themselves these large distances in such a small amount of time. They need, as a group, to secrete large amounts of biosurfactants, which are actually the same rhamnolipids that Thomas spoke about. So they need to secrete these large amounts of biosurfactants. They need to propel themselves using flagellars, and then they travel towards the edge of the plate. So we're very interested in using this as a model system to understand group behaviors in Pseudomonas aeruginosa. So we can turn off the movie now, and we can go to the next slide. This slide here shows a very simple experiment that we did just to demonstrate that swarming across surfaces has a huge advantage for a population of Pseudomonas aeruginosa. So we see here in this first bar here, we see that if we inoculate Pseudomonas aeruginosa in a petri dish and we let them swarm away, and then we scrape all the cells off at the end of 24 hours, and we count how many bacterial cells we had in that colony, we see that swarming bacteria can make up 17 billion bacterial cells. But if we grow the same bacteria in a different type of media, where we actually use the same nutrients, but we prepare the agar at a much higher percentage, so this is 1.5% agar as compared to the 0.5% agar that we use in soft agar, this makes it impossible for the bacteria to swarm, so they stay localized at the center of the petri dish, where they deplete all the nutrients around the colony. And that means they will grow much, much less, and we see that by the number of cells that this colony has. We can cause the same effect if we use soft agar, but now we, rather than using wild-type Pseudomonas aeruginosa, we have isogenic mutants. So mutants like the FLAG-K mutant, so it's a mutant that lacks a gene that's essential to form a functional flagellum. And the same thing here in this RAL-A mutant, which is a mutant that cannot produce the biosurfactants, the rhamnolipids that Pseudomonas aeruginosa needs in order to lubricate the surface. So these bacteria also cannot swarm, these isogenic mutants, they don't swarm, and again they deplete all the nutrients around the colony, so they grow much less. So this is just to show that if bacteria are allowed to swarm, they can travel larger distances, they can extract much more nutrients out of the agar surface. So swarming is definitely an advantageous trait from the point of view of the population. Now, as I've shown here with this plot, swarming requires several traits. I'm showing here two examples. So it requires flagellar motility, so the bacteria they need to have a functional flagellum in order to be able to swarm, and also requires the production and secretion of biosurfactants. And in addition to these, microbiologists have found more traits that are necessary to swarm in order for Pseudomonas aeruginosa to swarm. So what we asked in this study is what happens now if we apply a selective pressure for swarming. And given that there are so many different traits that the bacteria could evolve in order to swarm better, we argue that it's very difficult to predict the path that evolutionary adaptation would take. Okay, so we're asking, how would selection for swarming drive the evolution of Pseudomonas aeruginosa? And so we were inspired by experiments from evolutionary biologists, most notably Richard Lansky, who does long-term experimental evolution in E. coli. And so my postdoc, Dave Van Ditmarsh, he carried out these interesting experiments. So Dave is actually attending the webinar too. So hi, Dave. So first time that Dave carried out this experiment, he ran 3 independent lineages in parallel. And so what he did is he inoculated a Petri dish with Pseudomonas aeruginosa. He let the colony swarm, and then at the end of the day, he scraped all the cells off into a test tube, shake that tube, and then used a small fraction of that to inoculate a fresh plate. And he let those bacteria swarm, he scraped the cells off, and he did this over and over in the course of several days. Now, the nice thing of this experimental evolution is that you can also store the intermediary stages. So, for example, at the end of each day, we can keep the liquid media here. We freeze it in a glycerol stock, and that constitutes what Richard Lansky calls the frozen fossil record. So that means anytime we see something happening in the future, we can go back in time and take these glycerol stocks and resurrect our bacteria to study what happened. So it's a great advantage when you're studying experimental evolution with bacteria as compared to studying, for example, evolution in animals, where you have to rely on actual fossil records. Here we keep all the fossil records, so we can go back in time and study these bacteria again. Okay, so as I said, when we first started this experiment, we didn't know what to expect. So we started 3 independent replicates, so 3 parallel lineages, and this is how the bacteria look like at the end of day 1. So you see in all 3 replicates we have normal swarming motility with the typical branching patterns that I showed you before. So they've scraped these bacteria, he used them to inoculate day 2, and this is the end of day 2. So again, we have normal swarming. I'll go cycling through days now. This is day 3, so still normal swarming. Day 4, and at day 5, we saw that replicate number 2 had a different phenotype. So it looked like it was covering a higher percentage of the plate, right? So we continued the experimental evolution. This is day 6, day 7, day 8, and by day 9, we saw that all 3 replicates had evolved this ability to cover the entire plate, and we call this phenotype hyperswarming. Okay, so this is a summary of our experiments here. We have the different passages in each column, right, 2, 3, 4, etc. And in each row, we have the different replicates. We saw that replicate number 2 evolved hyperswarming at day 5, and then the other replicates evolved hyperswarming later. Okay, so this brings up very interesting questions. The first question that we had in mind is this hyperswarming a heritable phenotype? Meaning, can we go to the hyperswarming plates, like the ones that we highlighted here with different colors, and can we isolate clones that are by themselves hyperswarming? Okay. Now, the second question that we had in mind was, given that we saw hyperswarming evolve in 3 independent lineages, are they all going to harbor exactly the same mutation, or rather is this an example of convergent evolution where we see bacteria evolving a similar phenotype but through completely independent molecular pathways? And again, at this time, with just these data, we had no idea what to expect. Okay, so to answer question number 1, we went into this place that we highlighted here with different colors, and we were capable indeed of isolating different hyperswarmers. So we actually have here a video. It's this video called Hyperswarmer, and we're going to play that one now. And just to show you again a time lapse, but in this case of a hyperswarming clone that we isolated, just to show you how different the phenotype is compared to the wild type. So now we see that the bacteria do try to form tendrils, but then they're capable of covering the space in between tendrils. So that was quite interesting to us, and especially to see that this evolved independently at least 3 times, right? So we can close the video, go back to the presentation. Okay. So what we did next is we looked at hyperswarmers that we evolved independently from different experiments. Some of them were coming from same experiments, and this is what these different colors here mean. So these are clones, all in yellow. They were isolated from the same plate. Green, the same thing, and cyan, the same thing, and magenta. And we measured a battery of phenotypes. So we measured their ability to swim, to twitch, to form biofilms using a crystal violet assay and the supernatant in those biofilms, and their ability to secrete rhamnolipids. So we just measured these different phenotypes to try to get an idea of whether the different clones that we evolved independently, whether they were all the same mutants, or rather if they had different phenotypes. And what we saw was indeed that we had mutants that had diverse phenotypes. So this was suggesting that the mutants were not all harboring the same mutation. Now, this also allowed us to pick representative mutants from each of these clusters of phenotypic evaluation, and we carried out whole genome sequencing of these clones to try to find what were the mutations that were causing them to become hyperswarmers. So we sequenced all these clones that are here highlighted, boxed out in black, and we also sequenced our wild type, and we compared their genomes, and we indeed were capable of finding mutations that explained hyperswarming. So these mutations, they were all single nucleotide changes in the genome, so that was very interesting. And we found 2 types of mutations, both encoding for amino acid substitutions. And also what was interesting is that both mutations fell on exactly the same gene. So even though the mutants isolated from different experiments had diverse phenotypes, they were mutants from the same gene. And the gene where these mutations fell is a gene called FLIAN, which in Pseudomonas aeruginosa is known to regulate the number of flagella. So that's quite interesting. Let's see what else FLIAN does. So the protein encoded by FLIAN interacts with 2 other proteins, FLIQ and FLHF, which are both involved in constructing flagella in some way. So FLIQ is a master regulator that then sets off the synthesis of the flagellar units, and FLHF is a molecule, is a protein that localizes at the pole of Pseudomonas aeruginosa and directs flagellar assembly there. Now, what happens when we knock out FLIAN, and this is what they did in this original paper here where FLIAN was discovered. They saw when they knocked out FLIAN, Pseudomonas aeruginosa went from having a single polar flagellum, which is what the wild type has, single polar flagellum, and goes to having multiple polar flagella. Okay. Now, what's interesting is when you knock out FLIAN, you have bacteria that have multiple polar flagella, but they cannot swim. So we don't know exactly why this happens. The flagellar bundle is not functional. Now, in our case, what we were doing when we evolved hyperswarmers is that we had bacteria that were indeed multiflagellated, like what we see here. So this is actual electron microscopy imaging from one of our hyperswarmers. They are multiflagellated, but unlike the knockout, these bacteria actually swim better than the wild type, whereas the knockout cannot swim. Our hyperswarmer mutants have multiple flagella, and they swim better than the wild type. Okay, so that was quite interesting because it was suggesting a mechanism by which hyperswarmers were outcompeting the wild type through single point mutations. Now, once we found that the cause of hyperswarming was point mutations in FLIAN, we went back to our clones and we sequenced FLIAN in all the mutants that we had isolated, and we confirmed that indeed all of them had point mutations in the gene, and it confirmed also the clustering organization of our clones by phenotypes. We also conducted more experimental evolution, so we went back to the lab and we ran the experimental evolution many times more, and we found that all the hyperswarmers that we could isolate, all of them had point mutations in FLIAN. All of them had single nucleotide changes in FLIAN. So that was quite interesting. We saw one of the residues, particularly residue 178, tends to get mutated more times. Hyperswarmers that have that gene mutated are more frequent than others, but we found other mutants as well, and they all had point mutations in this gene. Okay, so we also went back to the lab, and we confirmed using microbial genetics that the hyperswarming mutations are both necessary and sufficient for hyperswarming. So here we took the wild-type bacteria, and through genetic engineering, we introduced the mutations, and we obtained hyperswarmers for both of the type of mutations that I showed you before. And we also did the reverse experiment where we took evolved hyperswarmers, and we reverted their FLIAN sequence back to the wild-type FLIAN sequence, and we obtained the wild-type swarming phenotype. So this type of experiment shows beyond any doubt that the mutations are indeed necessary and sufficient. Okay, so how does hyperswarmer win its competition against the wild type? Whenever we're conducting experimental evolution and we're doing these serial passages, we're basically buying time until spontaneous mutations hit a certain nucleotide in FLIAN that confers an advantage. Now, that advantage is not a growth rate advantage, and this we saw by growing the hyperswarmer clones in a growth curve assay, where we can measure very precisely their growth rate in liquid. So this is not in swarming plates, but it's actually in liquid culture, which is completely mixed. So we're evaluating the ability of planktonic cells to grow, and we saw that the hyperswarmers, they do not grow faster than the ancestral wild type. They actually grow slightly slower. So whatever is conferring the advantage to hyperswarmers is not simply a faster growth rate. Now, to show where their advantage was coming from, what we did next is we labeled bacteria with different fluorescent markers. So we have here, just as an example, Pseudomonas aeruginosa wild type labeled with GFP, so the colony is all green. Here we have the same Pseudomonas aeruginosa wild type labeled with DS red, so the colony is all red. And here we have a mixed colony where we mixed a GFP labeled wild type with DS red labeled wild type. And you see that the colony is yellow throughout, which means that the green and the red are mixing very well in this colony. Okay. Now, when we conducted the same type of labeling experiments, but we mixed the wild type labeled in red against a hyperswarmer labeled in green, we see that they no longer mix very well, but rather we see that the hyperswarmer tends to localize at the edge of the colony. And we can see that here, for example, when we mix wild type with hyperswarmer at a ratio of 1 hyperswarmer per every 100 wild type, we see the hyperswarmer here in green localizing at the edge. Here it's for a ratio of 1 to 10, so 1 hyperswarmer per 10 wild type. We see, the edge really enriched on hyperswarmer, and here if we mix them both at 1 to 1, we don't even see the wild type anymore. It's just completely outcompeted by the hyperswarmer. Okay? So this is suggesting a mechanism by which hyperswarmers are winning the competition. By gaining point mutations that confer them an advantage in terms of spreading on the surface, they can reach to the edge of the colony where they gain prime access to fresh nutrients as the colony expands. Now, I think this was also very interesting from a basic science point of view because we're actually looking at a single step in an evolutionary path towards a fitter individual. And it's a nice example of experimental evolution because it shows how point mutations can confer fitness advantages. In this case here, we're not evolving a whole new flagellum from scratch. We're simply evolving the ability of the bacteria to synthesize more of their flagella. So it's like a very, very simple step in their climb towards a higher fitness peak. Given that it's so easy for Pseudomonas aeruginosa to go from having a single flagellum to having multiple flagella, all it takes is to mutate a single nucleotide. We are also interested in the question of why is Pseudomonas aeruginosa monoflagellated to begin with. So why is it monoflagellated in nature? So what we did here is we acquired a library of both environmental isolates of Pseudomonas aeruginosa, as well as clinical isolates that we obtained here from the infectious disease clinic at Memorial Sloan Kettering, and we measured their swarming phenotypes. Here we're evaluating in a swarming assay how much of the plate is covered by the colony, and here in the y-axis how round does the colony look. So this is a measure of roundness of colony. If that value is very high, that means the colonies are very, very round. If that measure is low, it means the colony is branched, like in the wild-type swarming. So this is our wild-type Pseudomonas aeruginosa PA14, sits right here in the phenotypic space. And if we look at all the clinical and environmental isolates, we see that they span a wide range of phenotypic space of swarming colonies. But our hyperswarmers, they sit way out here, so in a completely different phenotypic space. So there's nothing in the clinical and environmental isolates that we looked at, there's nothing that resembles our hyperswarmers, which really suggests that in the lab we provided specific conditions that would probably not favor, would not be favored when these bacteria are in the wild. Okay, so we asked the question, if having multiple flagella confers such a huge advantage, why don't we see it in the wild? And one explanation is that hyperswarmers are actually becoming specialists at doing something very, very well. So they're becoming specialists at outcompeting their ancestral in swarming plates, but that specialization can come at a trade-off of doing some other phenotypes really badly. And one phenotype that we know is essential, both in the environment and in the clinic, is biofilm formation, as Thomas very nicely put. So we had seen before when we evaluated our phenotypes that hyperswarmers are actually much worse than the wild type at forming biofilms. So what we asked next is, in a mixed biofilm, where we have both hyperswarmers and wild type competing in a mixture, can the presence of the wild type compensate somewhat for the loss of biofilm formation by the hyperswarmer? And the answer to that question was no. So although hyperswarmers can outcompete wild type very well in swarming competitions, when they're facing the wild type in a biofilm competition, then they're completely outcompeted. And here's data showing that. Here we mixed ancestral and wild type at different ratios, and we see the ancestral always increases in frequency, whereas the wild type decreases in frequency, to really demonstrate how they have a fitness disadvantage in biofilms. Okay, so here I'm showing again a picture from confocal microscopy. Here we mix the wild type and the hyperswarmer at different ratios. The wild type is labeled in red, the hyperswarmer is labeled in green, and we see after 20 hours that the hyperswarmer is severely outcompeted in the biofilm. Can we please play the movie again? Okay. So the movie is actually just to make a better point about how much the hyperswarmers are outcompeted. It's a 3D rendering coming from a confocal microscopy imaging of this mixed biofilm. So you see the hyperswarmer, which is in green, is completely outcompeted. Okay, so we can go on now to the next slide. Okay, so just in conclusion, what we showed is that repeated rounds of swarming motility select for hyperswarmers. Interesting, all hyperswarmers, they evolve through single amino acid substitutions in the same flagellar synthesis regulator, which is called FLIAN. All the FLIAN mutants that we isolated, they were hyperflagellated, and they were better swimmers than the wild type, so they can travel faster in a swarming colony, they can travel faster towards the edge of the colony, and they gain their competition by gaining prime access to nutrients. But then we also saw that the hyperswarmers, they have a huge disadvantage in biofilms, and this explains why we don't find hyperswarmers in nature, both in environment and in clinical isolates. So, as I said, this was published very recently in Cell Reports just last month. Our conclusions also suggest new insights that might be relevant for anti-biofilm therapy. As Thomas also mentioned, biofilms are a huge problem, and certainly biofilms of Pseudomonas aeruginosa are very problematic because they tend to be much more resistant to antibiotic therapies. And what our experimental evolution shows is that there seems to be this evolutionary trade-off between the ability of bacteria to move and their ability to stay put and form biofilms. In our case, the FLIEN mutations that we obtain in our hyperswarmer experimental evolution, they increase motility, and they do so at the expense of biofilm formation. So we tilt this natural balance between the two types of surface behaviors in Pseudomonas aeruginosa, and this could potentially lead to new therapies that specifically target genes such as FLIEN to reduce biofilm formation in pathogenic bacteria. Okay. So in conclusion, I just want to thank my lab. So this was the composition of my lab at mid-2003 last year when we actually finished the study. And also we have additional projects in cancer biology, bacterial pathogenesis, and in the study of the diverse microbial community that colonizes our intestine, the gut microbiome. And I also must thank our funding sources, the NIH, through the program's New Innovator Award and the Integrative Cancer Biology program. So thank you very much.
Excellent. Thanks so much, Joao. Finally, we have Aaron Mitchell. Aaron is an expert on fungal pathogenesis. His work focuses on how the major fungal pathogen, Candida albicans, can form biofilms and the gene regulation that occurs in biofilms during infection. Today, he will talk to us about Candida albicans biofilm formation. Thanks for joining us, Aaron.
Hi, thanks very much for the opportunity to speak to your audience, my audience now. And so let me open by saying that although it's well appreciated that bacterial biofilm formation ability is essentially universal, it turns out that among fungi, the biofilm formation ability is extensive as well. So this is a fungal phylogeny based on whole genome sequences from a few years ago, and while it's partial, we can see that both the basidiomycete and ascomycete domains are represented. And among these, the basidiomycete pathogen, Cryptococcus, forms biofilms. The familiar baker's yeast, Saccharomyces cerevisiae, forms biofilms. The emerging pathogen, Trichosporon, forms biofilms. The dimorphic pathogen, Coccidioides, that causes valley fever, that forms biofilms. Pneumocystis, which is not capable of long-term growth in vitro, actually grows better as a biofilm than in planktonic culture. Aspergillus fumigatus, a lethal pathogen, is able to grow as a biofilm. And finally, the subject of my talk, Candida species, are able to grow as biofilms as well. And this is actually the original micrograph, to my knowledge, of a Candida biofilm that had grown on a venous catheter in a patient, the pioneering work of Bill Costerton. So fungal biofilms have broad implications. They are implicated in device-associated infection, such as venous catheters and coronary stents. They are the growth form that creates mucosal and superficial infections on all kinds of tissue surfaces. They're also implicated in the commensal interactions that allow many fungi to exist peacefully in our bodies or on our bodies. And finally, they're implicated in mating reactions, where a large population of cells can get together and fuse. They're similar to bacterial biofilms, fungal biofilms, in many ways. For example, there's extracellular matrix material that's produced by the microorganism itself. There's high levels of antimicrobial resistance associated with every kind of fungal biofilm ever observed. They are sensitive to quorum sensing molecules, and they can impact both biofilms both quantitatively and qualitatively. And there's considerable cell heterogeneity, as you'll see in just a few moments. That's quite obvious. On the other hand, there are some differences between bacterial and fungal biofilms that cause us to rethink some of the paradigms, at least, that we apply to studying biofilms. Fungal cells are non-motile for the most part, and so the biological dissolution of biofilms by adding quorum-sensing molecules, pre-existing biofilms, that is, it's quite unlikely. And similarly, the structure of a biofilm reflects its initial assembly and is not modified at later times, as far as we know. The physical size of some of these biofilms, fungal biofilms, is considerable. For Candida albicans, the depth can exceed 500 microns, and we try to avoid that because it creates imaging problems in the lab. And then finally, there's considerable functional redundancy among adhesins, and this is a common theme now among many fungal species. So the subject of the rest of my talk is the fungus Candida albicans, and it grows in many different forms, but it'll be familiar to many of you as these two forms, the yeast form, which is about a 5-micron oval cell that grows by budding, and long tubular hyphae, which can exceed several hundred microns in length. And they're tubular cells attached end to end that grow by tip extension. So we grow biofilms in the lab on medical silicone, as shown here in this low-resolution scan EM, and you can probably make out the hyphal filaments even at this magnification. In the confocal, we can get quite much higher resolution, and especially using an embedding technique that my colleague Fred Lonnie devised here at Carnegie Mellon. So here's a side view of the biofilm, and you can probably make out the hyphal filaments pretty well. But we're going to take a few images of apical views through the biofilm so you can see what the structure actually looks like. So down at the bottom, we see that there's primarily a yeast layer of cells. It's quite dense, and there's a little bit of extracellular matrix material there. A little bit higher up, we have transition to hyphae, and density, overall cell density drops, and there is some matrix material as well. A little higher up, we have primarily hyphae, low density, not much matrix here. And then finally, at the top of the biofilm, we have the reappearance of little clusters of yeast cells. And those yeast cells are thought to be the cells that are released to cause dissemination from a biofilm. So biofilm formation, the process of biofilm formation actually reflects the structure based on lovely work from Julia Douglas and Mahmoud Danoum. So initially, yeast cells adhere to a device or a surface. And then as they proliferate, they start to acquire biofilm-associated phenotypes like the production of hyphae and high-level drug resistance. As the biofilm matures, there's accumulation of extracellular matrix material. And finally, those apical yeast cells are formed, and they're released to seed new sites of infection or biofilm formation. So what I'm going to focus on for the rest of my talk is a transcription factor, BCR1, that we identified that we knew governed an early event in biofilm formation. And as it's been studied over the years, we've come to understand that its functions are really integrated into many aspects of biofilm formation. And I think there are a few lessons there for us to think about as we analyze more fungal biofilm regulators. So BCR1, biofilm and cell surface regulator, is a C2H2 zinc finger protein. It was discovered through a systematic transcription factor mutant screen in my lab. And its major targets include cell surface protein genes and adhesin genes. Its significance has mounted over the years. It's required for biofilm formation in vitro under many different conditions. It's required in vivo in a lovely catheter biofilm model devised by my friend David Andes. It's required in vivo for a denture biofilm model, also developed by David Andes. And then it's required in vivo for both oral and vaginal mucosal infection models. And finally, its Candida parapsilosis ortholog is required for biofilm formation in that organism. So there are a few lessons that we've learned from these many years of study of BCR1 in my lab and in many others. And the first is that the functional targets reflect the assay. So in my lab, we first identified several cell surface adhesins as the major genes that respond to BCR1 activity. And so we reasoned and then documented through measurements that BCR1 governs cell substrate adherence and cell-cell adherence. And that made perfect sense if its major targets were adhesion genes. But in David Saul's lab, they took a completely different approach in which they looked for BCR1 targets that might govern biofilm matrix integrity, and indeed they found those. And they found a gene family called the CFEM protein genes that had not been known to function in biofilm formation previously. So the second lesson that we've learned is that the expression targets actually reflect the environment. And this is important when we think about which pathways might be required for various kinds of biofilms to form. So one lovely study comes from Geraldine Butler's lab in which she compared BCR1 responsive gene expression in cells grown in two different media. And what she found was that the overlap between the two data sets was really minimal and quite a surprising result compared to at least our bias previously. And then in my lab, we compared BCR1 mutant and wild-type strain grown in vitro and grown in an oral infection model in collaboration with my friend Scott Filler. And we looked at genes that were up and down regulated in the mutant compared to the wild-type as reflected in this heat map. And what you can see is that the vast majority of genes that we assayed responded to BCR1 either under in vitro conditions or in the oral infection model, but very few genes responded under both conditions. So we can explain the BCR1 target variation through recent studies from Clarissa Nobley and Sandy Johnson's lab that have revealed that there's considerable convergence of biofilm regulators in terms of their targets. And what they did was to take a systems biology approach to look at the targets of all known biofilm regulators. And what they found was that many target genes are shared, as illustrated in this diagram, in which yellow, blue, and gray dots represent responding genes and the transcription factors that govern their expression, or their names are written out. So you can see that it's easy to picture that BCR1 target genes that might be affected in one environmental condition would be non-responsive to BCR1 under other conditions if, for example, some other biofilm regulator like EFG1 were expressed at higher levels. So the third lesson, and I think the most important one when we think about fungal biofilms, is the functional targets reflect the pathogen. And here I'm looking at work from Geraldine Butler's lab in which she has manipulated Candida parapsilosis, another major biofilm former, so that she can make mutations. And they made a disruption of the BCR1 ortholog in that organism. And as shown in these in vivo catheter images, the BCR1 deletion mutant is defective in biofilm formation. And that looks very reminiscent of the situation in Candida albicans, actually grown in the same catheter model in which a BCR1 mutant is defective in biofilm formation. But when the Butler lab went to look at the targets of Candida parapsilosis BCR1, she found that those CFEM protein genes, the ones that seem to participate in matrix integrity, are a common set of targets between the two BCR1s. But that adhesion genes, if they could be found, are very different in Candida albicans and Candida parapsilosis. So I'd like to just close with a couple of broader lessons about what we've learned from fungal biofilms and study of BCR1. And one is that functional redundancy is extensive. There have been overlapping functions found for transcriptional regulators of biofilm formation, adhesins, matrix pathways, and drug resistance mechanisms. And what I'd like to speculate is that because fungi have such a limited ability to restructure a biofilm, perhaps they have used redundant mechanisms to allow tolerance of environmental variation that bacteria could overcome by just picking up and moving to a new location, rebuild the biofilm in a more hospitable environment. And the second point that was somewhat surprising is that regulators are conserved and targets are not. And again, if we look at the work done in Candida albicans and the work from Geraldine Butler in Candida parapsilosis, we see that there are at least two shared biofilm regulators, BCR1 and EFG1. And there's really minimal conservation of their target genes when we look at the two species. And this makes me wonder whether perhaps the evolution of regulatory circuits with the multiple relevant inputs is a slow step. That is, these are highly tuned regulatory circuits, whereas the evolution of proteins that can recognize substrates or interact or promote cell-cell interaction, many of those have fairly low affinities for their targets. And so perhaps there's relatively rapid evolution of adhesions, target genes of these regulators. I'd like to close by thanking the many people over the past decade who have contributed to biofilm studies for my lab, notably Lindsay Zhu, Jigar Desai, Sarana Fanning, Jonathan Finkel, Clarissa Nobili, Shantanu Ganguly, and many fantastic collaborators I've had over the years, including Fred Lonnie here, David Andes, Scott Filler's group, Vinnie Bruno, and Andre Nantel. And my funding has come from the NIH, the Burroughs Wellcome Fund, Harold S. Ginsburg Professorship for a while, and now the Richard King Mellon Foundation. Thanks very much for your attention.
Thank you, Aaron. Really interesting. Thanks again to all of our speakers for sharing insights with us. We have time for just a few questions from listeners, which we have been busily sorting and reading. I think the first question comes from a few different people, and I'm going to paraphrase it as follows. We've heard throughout this hour about the way biofilms are protective for the organisms that live in them. We're wondering if some of you might want to talk a little bit about whether living in a biofilm offers some vulnerability that we might exploit. Let's see, should we direct that question to Thomas?
Yeah, I think it's a very, very good question. Just on top of my head, it's I can't really picture anything which would be a disadvantage of being a biofilm, except maybe some evolutionary perspectives, as we have talked about, but that you maybe could become inbred and could lose some of the traits that the wild type has. But then again, this development is probably to adapt to the certain environment that bacteria are in. So it's a good question. I'm definitely going to think about it, but I just can't think about disadvantages of being a biofilm.
Okay, maybe I can add a little bit to that question. I think part of the interesting fact of being in biofilms is that bacterial cells are living in an extremely crowded environment where they have to face the constant presence of the others and, of course, constant competition to do better than the others. And I believe that if we understand exactly what are these social pressures that the different bacteria are facing inside biofilms, we can eventually engineer new therapies that exploit these therapies. So my answer to that is pretty generic, but it's we should aim to find out what's so different about living in a biofilm as compared to living in platonic cells, and what are the different selective pressures involved in staying in the biofilm. Because we do know that when bacteria stay in biofilms long times, and they do this in the course of chronic lung infections in cystic fibrosis patients, for example, the bacteria do adapt to those environments, and they accumulate certain types of mutations. So if we understand exactly the microenvironment that the bacteria are experiencing in biofilms, this could potentially lead to new therapeutical strategies that, unlike antibiotics, do not select for resistance. So that's also part of my goal is in the lab, is to try to understand what are the selective pressures that drive bacteria to adapt in biofilms, and whether we can exploit these to turn bacteria against each other, since they're in such a very, very crowded environment, in a way that you possibly couldn't do if they were in planktonic mode. But that becomes extremely interesting in biofilms because the biofilms are actually the major cause of the chronic infection.
If I can just add one point, is that the biofilms are cells that are tethered together. So in some ways, if we could deliver antimicrobials to antimicrobial treatments to specific targeted regions, we might be able to wipe out a biofilm much more easily than using a systemic treatment of some sort for a bloodstream infection. And so the idea is that same kinds of approaches that might be applicable to wiping out solid tumors might also work for biofilms.
Nice. And our next question that we have is from someone who's asking whether or not there are mixed biofilms, so a heterogeneous composition of two or more bacterial cells coexisting together, and wondering whether or not this is observed in vivo and what the effect of a mixed biofilm is to antibiotics and overall survival of bacteria or fungi in the biofilm.
Maybe I could go first, Thomas, here at that. So some of my collaborators, they have been looking in soil for mixed biofilm. They see that a mixture of bacterial species can induce biomass formation. Also, they can promote each other. In vivo and in infections, most chronic infections are actually multi-species. But when we look actually at try to visualize the bacteria using FISH technique with different probes, different bacteria, we can find different species in the same infection. But it doesn't seem, at least in the majority of infections, it doesn't seem that the bacteria actually co-aggregate. In some they do, but in others they can be maybe 20 or 50 micrometers apart in the CF lung and chronic wound. But it doesn't seem that they intermix in these infections. So in nature, and when you have a natural microbiota, and when there's maybe a fight or nutrients sparse, then synergism is important. But in infections, I think there's not really a succession for collaboration, and so they kind of seem to be present in the same infection, but do not intermix. It's not necessary for them. It's not a driving force because the nutrients are not limited, etc.
Okay, we have just a couple more minutes, being generous. So let's have one more question. This comes from Aura Reyes, and the question is: would hyperswarming happen in other species of Pseudomonas, and would that be an advantage in applications such as bioaugmentation, bioremediation in environmental fields?
So I guess that question is for me. This is João. The short answer is we don't know yet. It's a very interesting question. Whether the hyperswarming phenomenon, just mutating single nucleotides in the gene FLIAN would also happen in other non-pathogenic forms of Pseudomonas. I'm very interested in that particular question more from a very practical point of view. I think the hyperswarming experiment is very interesting from the point of view of experimental evolution, studying evolution in the lab. And one of my concerns about not having more people working on this topic is that Pseudomonas aeruginosa is a pathogenic bacterium. So not all labs can start doing experiments with this bacterium. You need at least some level of safety. In the U.S., it's called biosafety level 2. If we were capable of doing this in a non-pathogenic environmental microbe, let's say Pseudomonas fluorescens, that could potentially open up the way for more labs to reproduce our work and to use it as a model system to try to understand evolution in the lab.
Great. All right. Thanks so much, João. And we really have to stop. Unfortunately, there's a lot of great questions here that we're not going to get to. But thank you for joining us today and for submitting some excellent questions.
We'd like to say thanks again to our speakers, Thomas Bjarnsholt, João Savir, and Aaron Mitchell for their interesting presentations, and/or BitPlane for their generous sponsorship, and of course, all of you out there for listening. The webinar will be archived on the CellPress website. Keep your eyes open for more CellPress webinars. Bye. Yep.